Hello, good students of Dr. Baliraine's General Biology class. Today, my group and I are going to talk about mRNA transcription and translation. I hope you enjoy. And now introducing our first major player, the RNA polymerase. Step is initiation. Promoters signal the initiation of RNA synthesis, like the Tata box. Transcription factors bound to the DNA help eukaryotic RNA polymerase recognize promoter sequences. And then, once it is bound, it starts to unwind the DNA. RNA polymerase moves along the DNA, continuing to untwist the double helix and separate the hydrogen bonds that hold the nucleotides together. It exposes about 10 to 20 DNA bases at a time for pairing with RNA nucleotides. It also attaches these RNA nucleotides together. And once it's done with that, it rewinds the DNA sequence. But it doesn't stop there, because it has to do it again. And again. And again. Elongation continues until the RNA polymerase finds a polyadenylation signal sequence in the DNA, which translates to AAUAAA in the RNA. The transcript is then cut 10 to 35 nucleotides after poly A site, and the RNA polymerase continues until it falls off. And thus we have a perfectly good mRNA strip. Not! The pre-mRNA has to be modified by enzymes, like the RNA ribozyme. Ribozymes remove introns, or non-coding regions of the mRNA. They also join together exons, which are the coding regions of mRNA. This is called RNA splicing. Other enzymes attach a poly A tail to the 3' end and a nucleotide head to the 5' end. And now we move on to translation, starring everyone's favorite, the ribosome! Everybody give a hand! First, a correct match between a tRNA and an amino acid done by an enzyme amino-acyl tRNA synthetase must be performed. There are 20 of these enzymes, one for each amino acid. First, the enzyme joins the amino acid and then the correct tRNA.
Note, this does require ATP.
Then the smaller subunit of the ribosome binds to the mRNA binding site on the 5' end. This is what we would consider initiation. Then the larger subunit comes to join the party. The larger subunit has 3 areas: the A site, the P site, and the E site. The A site is where a tRNA with an amino acid joins the ribosomal unit. The P site is where the polypeptide is attached to the rest of the chain. The E site is where the tRNA exits, hence its name, the exit site. Then begins elongation. The tRNA with an amino acid joins the mRNA at the A site, then is moved over to the P site, where the amino acid joins the already growing polypeptide chain. Then it is moved over to the exit site, where the tRNA pops off. Elongation continues until a stop codon, which is UAG, UAA, or UGA, is found on the mRNA. Then, the polypeptide pops off. And there you have it, folks. We have just described transcription and translation. Not with all the detail, but enough to get you to pass your test. I hope you all enjoyed the video, and check out the rest on my channel.